Imagine your doctor wants to test a new, a highly experimental cancer drug on you.
Right.
But instead of giving you the pill and just, waiting around to see if you get better or suffer terrible side effects, they boot up a computer.
Yeah, they run a simulation.
Exactly. They load a flawless digital replica of your exact cells. They drop the digital drug into the simulation and they just watch what happens and fast forward.
Which is incredible.
Right. If it fails, they try another one and another, all in a single afternoon. And if you're listening to this deep dive today, I want you to know that this sci-fi scenario just took a massive step toward reality.
It really did. I mean, it is a fundamental shift in how we approach disease.
Yeah.
Because for decades, computational biology has been stuck in what we call descriptive inference.
Descriptive inference. Okay, break that down for us.
So we look at a cell after we've already done something to it in a lab, and we just try to describe what happened. We're looking at the aftermath.
Like a detective at a crime scene.
Exactly.
Yeah.
But the holy grail is predictive simulation. We want to run high-fidelity experiments entirely in a computer, in silico, as we say.
Right.
We want to know exactly how a cell will react before a chemical is ever physically synthesized in the real world.
And the reason we haven't been doing this all along, right, is because it's just a brutal math problem.
Oh, the scale it's unimaginable.
Yeah, the biological landscape is impossibly vast. Like, if you want to predict how every conceivable human cell type responds to every potential new drug or, every genetic mutation, you are looking at a combinatorial explosion.
Absolutely.
There simply aren't enough hours in the universe or money in the global economy to run all those physical experiments in a wet lab. We need a shortcut.
Which brings us to the focus of our analysis today. It's this groundbreaking generative AI framework called AlphaCell.
AlphaCell.
Yeah. And the researchers behind it, they haven't just built another database. They have constructed they call it a virtual cell world model.
A virtual cell world model.
Right. It serves as a true digital twin for living cells.
Okay, but to really grasp how AlphaCell pulls off this, simulation magic, we have to look at the graveyard of previous models.
There's definitely a graveyard.
Because if you look at foundation models out there, things like SCGPT or STATE, they all stumble over the exact same 3 systemic flaws.
Right. They all hit the same walls.
And the first major flaw is how they handle data. They suffer from what the paper calls incomplete latent representation.
Yeah, which is a highly technical way of saying they take massive shortcuts to save computing power.
Right, because computing power is expensive.
Exactly. I mean, the human genome has over 19,000 protein coding genes.
19,000?
Yes. But feeding all 19,000 variables into an AI, it just requires massive computational resources. So earlier models just chop the data down.
They just delete it.
Pretty much. They only look at the top 1,000 to 2,000, what we call highly variable genes, or HVGs.
Wait, so they essentially throw away roughly 17,000 genes before they even start doing any math?
Yeah, they just toss them out.
That's insane. It's like trying to predict global weather patterns by only monitoring the 2,000 loudest thunderstorms.
That is exactly what it's like.
And you're completely ignoring the rest of the atmosphere. You might see the big, obvious events, but you are entirely missing the invisible, subtle pressure systems that are actually driving those storms in the first place.
That is the perfect analogy. By stripping out the low abundance genes, these older models completely ignore the master regulators.
Right.
Think of master transcription factors in a cell. They might not be highly expressed, meaning they don't shout loudly, but they act like the CEO of a company.
Oh, I see.
A CEO whispers a single order, and it changes what thousands of factory workers do. If your AI is only listening to the loudest workers, it completely misses the actual cause of the change.
And when AI misses the underlying cause, it just starts to guess.
Right, it has to fill in the blanks.
Which triggers the second major flaw, biological reconstruction distortion.
Yeah, the hallucinations.
Exactly. Because these older models are working with this heavily truncated, chopped up data, they spit out abstract mathematical predictions that simply do not translate back into full biological reality.
They literally hallucinate biology. The math might look really clean on paper, but it doesn't represent a real, viable cell.
And that ties directly into the third flaw, right?
Yeah.
The dynamic transferability deficiency.
Yeah, that's a mouthful.
It is, but it gets to the heart of how we model change. Existing models treat cellular changes as discrete, jagged jumps.
Right. They look at a cell and label it healthy at point A and sick at point B.
But biology isn't a light switch.
Exactly. It's a continuous fluid trajectory. Because older models only see the before and the after, they completely fail to learn the universal underlying laws of biological physics that actually cause the transition.
Okay, so let's just do a quick recap for you listening so we can lock this in before we move on.
Good idea.
So current models fail for three big reasons. One, they truncate the genome to save time cutting out 17,000 genes. Two, because of that missing data, they hallucinate fake biology. And three, they treat disease as a jagged jump rather than a continuous flow.
Spot on.
Think about the last time you tried to solve a complex puzzle with only 10% of the pieces. You know how frustrating that is. How much of the picture did you miss?
You miss almost everything.
Exactly. So AlphaCell changes the game by just refusing to take shortcuts. It processes the full entire genome, all 19,253 genes.
But taking in that much data, it requires a fundamentally new architecture. We need a place for these digital twins to actually live.
Right, which brings us to the first major pillar of AlphaCell, building the virtual cell space.
Yes, the encoder. To build a reliable simulation, you need a flawless mathematical coordinate system. So the researchers start with incredibly rigorous data pre-processing.
They clean up the data.
Very aggressively. They align every single piece of data to the HGNC standard, the UGO Gene Nomenclature Committee.
Okay, why is standardizing the names so critical here? I mean, a gene is a gene, right?
You'd think so, but historical biological databases are famously messy. One single gene might have 3 completely different abbreviations depending on which lab discovered it.
Oh, wow.
Yeah, which leads to massive semantic ambiguity for an AI. It gets confused. By locking onto the HGNC standard, AlphaCell creates a strictly bijective entity mapping.
Bijective, meaning one-to-one.
Exactly, one-to-one. Gene A is always gene A. There is 0 translation error.
Okay, so the map is standardized. We have clear labels. How do they actually measure what's happening on that map?
They look at the raw UMI counts.
UMI.
Unique molecular identifier. It's essentially a tiny barcode that tells us exactly how much a specific gene is expressing itself.
Okay.
But instead of just feeding those raw numbers straight in, AlphaCell uses a 100x fine-grained tokenization strategy.
100x.
Yeah, it maps those UMI counts into a precise vocabulary of 1,024 tokens.
Okay, let's use an analogy here for the listeners. Think of this like upgrading from a cheap old radio to a high-end stereo system. Older models might just have a volume dial that says loud, medium, or quiet. But AlphaCell's tokenization gives it a volume dial with 1,024 tiny distinct clicks.
Yes, it allows the AI to capture incredibly subtle gene dosage variations that older standard normalization techniques would just completely wash out.
And once that highly detailed, standardized data is ready, it's fed into the engine of Pillar 1. The encoder.
And this is where things get computationally fascinating. The encoder is a Mamba transformer hybrid.
Right.
And it's trained on a staggering 140 million single cells.
Okay, wait. I know transformers. That's the architecture behind things like ChatGPT, right?
Exactly.
They are incredible at seeing the big picture of a sentence.
Yeah.
But what is a Mamba transformer hybrid? Why combine them?
So transformers are brilliant, but they have a fatal weakness. It's called quadratic scaling.
Quadratic scaling.
Yeah. If you feed a transformer a sequence of 19,000 genes, the computational cost squares with every single addition.
Oh, so it just blows up.
It becomes impossibly heavy for the memory. Mamba, on the other hand, is a different type of architecture, a state space model.
Okay.
It acts like a highly efficient selective scanner. It processes the massive sequence sequentially, holding on to the crucial information while instantly discarding the irrelevant parts.
Without overwhelming the computer.
Exactly.
Yeah.
So by combining them, AlphaCell gets the deep contextual understanding of a transformer with the sheer speed and sequence handling power of Mamba.
And this hybrid engine has one primary job, right? It acts as a massive information bottleneck.
Yes.
It takes the expression data of all 19,000 genes and compresses it down into just 32 coupled state channels.
Right.
We are talking about shrinking the entire human genome into a dense 32 by 128 dimensional mathematical grid.
Which naturally sounds entirely contradictory to everything we just said.
Exactly. Let me push back here because I know the listeners are thinking this too. We just spent the entire first segment heavily criticizing older models because they threw away genes to save space. Now you're telling me AlphaCell takes 19,000 genes and aggressively crushes them down into a tiny 32 by 128 grid. How is that any different? Aren't we losing the exact biological detail we just fought so hard to keep?
It is a crucial distinction, I promise. There is a massive mathematical difference between truncation and compression.
Okay, unpack that.
Truncation, which is what the old models did, is taking a book, ripping out 90% of the pages and throwing them in a fire before you even start reading.
You physically lose the information.
Gone forever. Compression, which is what AlphaCell does, reads every single page of the book and boils the meaning down to a highly concentrated summary.
Ah.
The compression acts as an intelligent filter. It forcefully discards stochastic noise, things like lab equipment errors or random measurement dropouts, but it mathematically preserves the structural topology of every single gene.
Ah, okay. So it's not cutting off pieces. It's boiling the biology down to its purest concentrate.
Precisely. And the way they train this encoder to do that involves 2 stages. Stage 1 relies on masked language modeling. They randomly hide certain genes in the data and force the AI to guess what they are based on the surrounding genes.
Oh, like fill in the blank.
Exactly.
Yeah.
This forces the model to learn the fundamental syntax and grammar of gene co-expression. If gene A and gene B are always active together, the model learns that biological rule.
And stage 2 is where they bring in the heavy mathematical artillery to really clean the data.
Oh, yeah.
They use 2 specific mechanisms, a DDAN and an ArcFace head. Let's use a mnemonic to visualize the DAN, which stands for Domain Adversarial Neural Network.
Okay.
Think of DNN as a strict, heavily armed bouncer at a very exclusive club.
That's incredibly accurate.
Yep.
Because in biology, we have a massive problem with what we call technical batch effects.
What does that mean?
Well, a cell sequenced on a machine in Boston might look mathematically different than the exact same cell sequenced on a machine in Tokyo.
Just because of the machines.
Just because of different lab temperatures or slightly different chemical batches, it's noise.
I
So Dan the bouncer looks at the incoming data, spots those artificial lab signatures, and literally kicks them out of the club.
Rah.
It ensures that the only thing entering our virtual cell space is pure machine-agnostic biology.
And while DDAN is kicking out the noise, the ArcFace head is working simultaneously.
Right.
Imagine ArcFace as a powerful magnifying glass that forces different things apart. Its job is to maximize the mathematical distance between different cellular identities.
Yes.
It ensures that the mathematical representation of, say, a heart cell is pushed as far away as possible from a brain cell in this virtual space.
Exactly. Preserving their distinct biological identities so they never bleed into each other.
Okay, so let's do another quick recap for everyone.
Let's do it.
If we look at what Pillar 1 achieved, the AlphaCell-based model encoder compresses all 19,000 genes into a dense 32 by 128 virtual cell space. And it uses DNN and ArcFace to remove noise and sharpen cell identity. Can you picture this virtual space? It's a perfectly smooth map where a cell's location is defined purely by its biology, not the lab equipment used to measure it.
Exactly. But a perfect mathematical grid is, well, it's useless to a doctor.
Right. You can't prescribe a treatment based on a 32 by 128 matrix.
No, you cannot. We have to pull the biology back out of the math.
How do we translate that compressed concentrate back into a readable 19,000 gene cell?
This brings us to Pillar 2. The observation interface, also known as the decoder.
The decoder.
And this is where AlphaCell completely breaks conventional AI design. Usually the encoder that compresses the data and the decoder that unpacks it, they're roughly the same size.
A symmetrical system.
Right. But AlphaCell uses an architecture called the inverted pyramid. The decoder is massively disproportionate. It is a colossal 1.2 billion parameter network.
Wow. Wait, why the vast asymmetry? Why is it so huge?
Because taking a complex reality and summarizing it, that's relatively easy. Okay.
But taking a summary and perfectly recreating reality without hallucinating details, that requires an immense reservoir of knowledge.
Right, that makes sense.
So AlphaCell uses a mixture of experts, or MOE architecture, to handle this.
Let's explain MOE because it's a brilliant concept. Instead of having one giant sluggish AI brain trying to figure out every single gene.
Which would take forever.
Right. A mixture of experts routes the data to specialized subnetworks. Imagine walking into a massive hospital.
Okay.
You don't have one single doctor trying to perform brain surgery, fix a broken leg, and deliver a baby.
That would be a disaster.
Exactly. The front desk, which is the router, sends the brain issue to the neurologist and the bone issue to the orthopedist. The MOE assigns specific gene pathways to specialized AI experts within the network.
And this immense network acts as AlphaCell's deep biological knowledge base. It takes that tiny abstract state from the virtual space, runs it through the specialists, and flawlessly translates it back into the exact expression magnitude for all 19,000 genes. Think of it this way. The encoder is a brilliant composer who takes a chaotic 19,000 instrument symphony and compresses it into a simple, elegant sheet of musical notes.
I love that.
That sheet is our compressed math. But you can't listen to paper.
Right.
So the decoder is a 1.2 billion piece master orchestra. It can read that simple sheet of paper, route the notes to the perfect musicians, and perfectly play every single instrument without missing a single beat.
It guarantees that our digital twin matches physical reality perfectly.
Okay, let's pause and recap Pillar 2.
Sure.
The massive MOE decoder ensures the abstract math can be turned back into a high-fidelity, whole genome reality, completely preventing biological hallucinations. What stands out to you listening right now? For me, it's the asymmetry. It's the realization that unpacking biology requires vastly more computational power than compressing it.
That captures the necessity of the asymmetry perfectly. We can now compress a cell, and we can perfectly unpack it.
But a static cell doesn't teach us how to cure a disease.
No, it doesn't. We have to introduce time, change, and external forces. We need to see what happens when the digital drug hits the digital cell.
And this is the hardest challenge in the entire field, isn't it?
Oh, without a doubt. And it's because of a grim reality in biology. Single-cell sequencing fundamentally destroys the cell.
Like, really? It destroys it?
Completely. If you sequence a cell to see what it looks like when it's healthy, it's dead. You can never apply a drug to it and then sequence it again to see the result.
Oh, wow. I never thought about that. You literally cannot observe the exact same cell twice.
It is the ultimate blind spot. You only ever have unpaired data. You have a bucket of healthy cells and a completely separate bucket of cells that received a drug and were then destroyed to be measured.
So you never actually see the journey between the two buckets.
Exactly. To solve this, AlphaCell introduces Pillar 3, the dynamic physics engine. And it uses a mathematical framework called OTCFM.
OTCFM, optimal transport conditional flow matching. That is a massive string of jargon. Let's break it down into something we can visualize. Let's start with the optimal transport part.
Okay. Optimal transport is actually a really old mathematical concept. Imagine a massive pile of sand on one side of a yard and a large hole on the other side.
Okay, I picture it.
Optimal transport calculates the absolute most efficient way to move every single grain of sand to fill that hole, expending the least amount of energy possible.
Right.
In AlphaCell, the sand pile is our healthy cells, and the hole is the drug-treated cells. The AI uses dynamic algorithms to instantly pair up these completely separate populations, constructing the path of least resistance between them.
It essentially builds a mathematical bridge across the blind spot.
Yes, precisely.
In the second half, the conditional flow matching, that's how it travels the bridge, right?
Right. Instead of just teleporting from healthy to sick, it models the effect of the drug as a deterministic vector field.
A vector field.
Yeah. It physically flows the cell from state A to state B over continuous time. Think of it like mapping the currents of a river.
And this is where the model has to be incredibly careful, because it has to simulate thousands of different drugs.
Oh, yeah. If it tries to learn them all at once, the math collides and creates gradient conflicts. The AI just gets completely confused.
So how do they solve that?
AlphaCell, again, uses that mixture of experts routing system, but it injects the specific drug signal using ADL.
ADL, adaptive layer normalization.
Yes, along with joint attention.
Let's translate ADL for a second. How exactly does it apply the force of a drug without confusing the rest of the simulation?
Think of ADL as a localized weather modifier within our simulation.
Okay.
It isolates the specific perturbation, the specific drug, and applies it mathematically as a dedicated physical force. It completely separates the force, the drug, from the object, which is the cell.
And joint attention?
Joint attention allows the model to see exactly which specific genes that local weather system is impacting. It modifies the cell state mathematically to see where the river current takes it.
So, if we take a step back and look at this physics engine, the AI has literally learned the fundamental laws of motion for cellular biology.
It really has.
It treats a drug not as a simple on-off switch, but as a continuous physical pressure applied to a cell's structure.
Exactly.
Let's just do a quick recap here. The flow model overcomes destructive testing by using optimal transport conditional flow matching. It treats perturbations as continuous physical forces applied within the latent space.
Perfect summary.
Imagine, instead of teleporting blindly from healthy to sick, you can watch the entire continuous journey. You can pinpoint the exact millisecond a specific gene cascade fails. How does that change our understanding of a disease?
It changes everything. It's revolutionary. But, a theoretical physics engine is just an elegant piece of software until it is tested against reality.
Right. Does this complex machinery actually outperform the models that currently dominate the field? The gauntlet is thrown. How did AlphaCell hold up when they put it in the arena?
Well, the researchers tested AlphaCell in two massive arenas. The first test was compositional generalization.
Okay.
They matched AlphaCell against the state-of-the-art models like State, SCGPT, and Gears on massive real-world data sets. Things like Cyplex and Tahoe 100 meter.
Heavy hitters.
Very. But they added a twist. They forced the competing models to use all 19,253 genes.
Oh. They took away their ability to truncate the data?
Exactly. They leveled the playing field.
And what happened when the older models were forced to look at the whole picture?
They catastrophically broke down.
Wow.
They suffered from what we call the curse of dimensionality. Because they don't have the sophisticated Mamba compression or the Dan bouncer to filter noise, the sheer volume of 19,000 variables just completely overwhelmed their math.
So what did they do?
They just output noise. AlphaCell, on the other hand, thrived. It achieved massive leads in macro F1 scores.
Which, in plain English, means it was incredibly accurate at predicting exactly which genes would turn on, which would turn off, and by exactly how much.
It absolutely crushed the standard tests. But there's a final, much harder test.
Right. Zero-shot generalization.
Yes. This is the ultimate proof of a world model.
Explain zero-shot.
The researchers asked AlphaCell to predict the biological response of a cell type perturbed by a drug that was completely absent from its training data.
Wait, completely absent. The AI had literally never seen this specific combination in its life.
Never. Not once.
How do the older foundation models perform when you ask them to predict something they've never seen?
This is where their reliance on highly variable genes, the HVGs we discussed earlier, reveals a fatal logical flaw.
Oh, I see where this is going.
If you have never seen a specific cell react to a specific drug, it is mathematically impossible to know in advance which of its genes will end up being highly variable.
Right. You can't know the loudest workers if they haven't started shouting it.
Exactly. You're entirely guessing in the dark. But because AlphaCell maps the entire genome and understands the underlying physics, it doesn't have to guess.
It just runs the simulation.
Yes. It achieved a multi-fold increase in Pearson correlation over the foundation model State. In some metrics, it was up to a 10-fold increase.
Let's explain Pearson correlation simply for the audience. It means the shape and magnitude of AlphaCell's prediction perfectly mirrored the shape of the actual real-world biological result.
Spot on.
And here is where the physics engine concept truly lands. Because AlphaCell learned the universal gravity of how drugs interact with gene networks in its virtual space, it can accurately predict how an apple falls on a planet it has never visited.
I love that analogy, yes.
It understands the biological laws of motion. So the unseen data isn't a mystery, it's just a new set of variables plugged into an equation it already knows how to solve.
That is the defining characteristic of a world model. It hasn't just memorized massive amounts of training data, it has abstracted the dynamic laws of biology. It generalizes to the unknown.
Okay, final recap for this section. AlphaCell vastly outperforms competitors in predicting unknown cell-drug combinations, proving it learned universal, transferable laws. If you are listening to this and digesting what we've covered, does this shift your view on what AI is capable of?
It really should.
We aren't just using algorithms to categorize pictures or write emails anymore. We have built an engine that runs reliable, continuous simulations of completely unseen biology.
We went from a noisy system that just took static, chopped-up snapshots of cells to a mathematically rigorous digital twin, one that filters out lab noise, maps all 19,000 genes, and applies continuous physical laws.
The implications for the speed, cost, and scale of therapeutic discovery, they cannot be overstated.
No. We could filter out millions of dead-end drug compounds in a simulation before ever spending a single dollar on wet lab testing.
So what does this all mean for you and for the future of medicine? I want to leave you with a final thought to mull over. Right now, AlphaCell maps the transcriptome, the genes.
Right, the RNA.
It is a master of the DNA blueprints. But imagine the near future when this virtual cell world model inevitably incorporates multimodal layers.
Oh, absolutely.
I'm talking about adding proteins, epigenetics, metabolism, the entire biochemical stack. If we can accurately simulate how one single cell reacts to a novel drug or an unseen virus today,
Right.
what happens when we chain billions of these virtual cells together?
You simulate an organ.
Exactly. Could we one day simulate an entire human patient's response to a cure? Could your doctor run 1 million virtual clinical trials on your personal digital twin, finding the one perfect bespoke treatment without ever putting your actual body at risk?
It moves medicine from a game of statistical probabilities into an era of perfect predictive vision. It really is the future.
The digital petri dish is here. Thank you for joining us on this deep dive. Keep questioning the blueprints of the world around you, and we'll see you next time.